All right, good morning, everyone. Welcome to day 2 of Human Cell Atlas Asia meeting, and we're excited about all the lineup and speakers for today. And importantly, we have a very distinguished speaker this morning opening up the session. And I hope, Alex, you're now online and can hear me. So before you start, I'd like to give a just brief introduction of the achievements that Alex has made and his interest in research. So Alex Shalek is a core member of the Institute of Medical Engineering and Sciences, an associate professor of chemistry and extramural member of the Koch Institute of Integrative Cancer Research at MIT. He's also an institute member of the Broad Institute, a member of the Reagan Institute, an assistant immunology at Mass General Hospital, an instructor in health sciences and technology at Harvard Medical School. So Dr. Shalek also received his bachelor's degree in summa cum laude from Columbia University and his PhD from Harvard University in chemical physics under the guidance of Hong Kong Park, and performed postdoctoral training under Hong Kong Park and Aviv Regev at Broad. His lab research is directed towards the development and application of new approaches to elucidate cellular and molecular features that inform tissue-level function and dysfunction across the spectrum of human health and disease. Dr. Shalek and his work have received numerous honors, including NIH New Innovator Award, a Beckman Young Investigator Award, a Cyril Scholar Award, a Pew Stewart Scholar Award, Avant-Garde DP1 Pioneer Award from the National Institute of Drug Abuse, and Alfred Sloan Research Fellowship in Chemistry, as well as in 2019 to 2020, Harold E. Egerton Faculty Achievement Award at MIT. And last but not least, a 2020 HMS Young Mentor Award. So it's a tremendous honor and privilege to have Dr. Shalek here to talk about his research. And he will talk about does cancer cell state matter? And we really look forward to his presentation today. So, Alex?
Thanks, Jay. That wasn't a little over the top or anything. Would have been great if you were just like, hey, this is Alex. But I'm so glad that I can join you all virtually today. Can you hear me?
Yes, we hear you. Fantastic. Please.
Great. Awesome. Thanks, Jay, for that introduction where I very much appreciated, would have also been very happy if you just said, hey, this is Alex. So it's a pleasure to join you all virtually. I'd be lying if I said I wouldn't prefer to be there in person with you, but I'm glad that I get to be with you here virtually today at HCA to talk a little bit about some of our recent work. But I really wanted to focus on discussing the importance of cell-stating cancer. And, we spend a lot of time thinking about the importance of DNA genotype in cancer and thinking about its relevance for druggability. And I want to try and convince you today that RNA phenotype is important as well. So before I begin, I just want to disclose that I have a series of financial relationships, none of which are relevant today, but for transparency, I list them here. Now, before I dive into our work on cancer, I wanted to start off by introducing what our lab is interested in broadly, and that's the idea of homeostasis or balance in our tissues. Now, this idea of homeostasis or equilibrium is a really simple concept. For example, I teach it to undergraduates at MIT, where in a chemical reaction, I could tell you what equilibrium is under different conditions. But when I try and think about what it is in our tissues at a cellular and molecular level, it's really hard to actually figure that out. But we know how important it is. If you have overexuberant immune responses, you can get tissue damage and death. And if you have too little of an immune response, you can become susceptible to opportunistic infections and the outgrowth of tumors. What our lab is really interested in is understanding what constitutes balance in our tissues. What are the deviations that occur under different perturbations? And how could we rebalance the system to restore human health or buffer it in the first place? To try and address these questions, what we like to do is to develop and apply tools that help us understand what's happening in these complex multicellular ecosystems that are our tissues. So what that means is, like many people there in the audience, we like to think a lot about the cells of which our body is made, to think about the diversity of different cell types, the qualities they possess, how broad they are and what they might target if they're immune cells, where they exist, how durable they are, all the same things that you're probably asking yourself. Now, the reason it's been such an incredible time to think about cellular biology over the past 5 to 10 years, as you all know, is that there have been these transformative advances in cellular profiling, where now instead of just having to look at one RNA or one protein, we can actually look pretty comprehensively. We can look at the genomic sequence of a cell. We can measure its chromatin accessibility. We can measure dozens of proteins, or we can even look at the entire transcriptome at once. Now, the question that might arise for you, as often arises for us when we think about this, is when we can measure so many different things, what's most important, when, and why? And so I wanted to talk about this a little bit in the context of cancer. I wanted to focus on this project that we've been doing through the generous support of the National Cancer Institute. This is part of a Cancer Systems Biology Consortium, or CSBC, that's really been focused on the idea of trying to understand mechanisms of drug resistance in cancer, both cell intrinsic and extrinsic. And it's been a series of projects where we brought together a number of different single cell technologies, some of which you might be very familiar with, like single cell RNA sequencing, but other ones to measure like mass and growth rate and functional assays and nanowells, along with biopsy specimens and models and some advanced computational methods. And I want to highlight that this is not work that we did independently. We're very collaborative, and this is something that could not have happened without being part of the big ecosystem that brought together individuals with expertise across disciplines. So we did this with Scott, Doug, and Chris at MIT, and David, Andy, Bill, and Brian at the Dana-Farber. And when I say we did this, what I really mean is that there's an incredibly talented set of students that push this forward. Peter Winter, who's a postdoc, Andrew Navia, who was a graduate student, has now graduated, and Mark and Tree, who now have their own labs at the Farber. And it's not just 4 people that it takes to do this work. It really is an entire village, and none of this could have happened without all of these incredibly talented graduate students, clinical coordinators, physicians, and the like that came together to make this happen. So what I wanted to focus on today is just one of the projects that we did here that really tried to understand cell extrinsic mechanisms of drug resistance. And so, as you're all aware, sort of the cornerstone of our precision medicine pipelines are DNA mutations. If somebody was to say to me today that I had a tumor, I would say, well, I want to take it out. I want to go sequence it. I want to figure out what the mutations are. Maybe I want to do it at a single cell level so that I can figure out all of the clones that exist and try and figure out if there's a drug that might work for my particular clone. I might need to go build some models, whether they're cell lines or organoids or PDXs, so that I could test whether or not the drug worked. And so, in a standard pipeline in my lab or maybe in your lab, what you might do is you might get a biopsy that has a KRAS mutant or an EGFR mutant or that's wild type. You go and you build a representative model, whether it's a cell line, an organoid, or the like, and then you go and sequence to confirm that you, maintain the mutation. And once you figured out that you did, you would want to go screen a large library of small molecules or do some genetic perturbations using, say, CRISPR-Cas9 to try and figure out what the vulnerabilities of that cancer was. Now, this works incredibly well when you have a mutation that's druggable and it's preserved in models. So if you have something like a BCR-ABL fusion where you can go in with ponatinib like you might see in ALL, but what do you do when this doesn't happen, when you don't have an actual genetic biomarker? And unfortunately, this is the case more often than not in cancers. And even where we have actionable mutations, very often, we evolve resistance to them. So a particular cancer that's a really good example of this is pancreatic ductal adenocarcinoma, or PDAC, which is incredibly devastating and challenging to detect cancer. When it's detected, it's usually already pretty far along. It requires surgery, and even with surgery, prognosis is pretty poor. But what's interesting about PDAC is when you look at the genetics, they're relatively uniform. Over 90% of patients have KRAS mutants, 75-ish% have P53 mutants, many have SMAD4 and CDKN2A mutations. And they're really, despite this relative uniformity, aren't very good genotype-directed therapies. There was recently this drug that was described that works for 1% to 2% of all KRAS mutants. It targets this G12C mutation, and that was a big deal. So you might say, if genetics haven't worked, what has the field really turned to? Well, what we've started to look at is RNA expression. And so people have done bulk expression profiling of tumors. And here I'm showing you vertical columns, which are tumors. And I'm showing you genes that correspond to one program called basal, which is more stem-like, and another program, which is classical, which is more epithelial-like. And what you can see is that there are some tumors that appear to be basal, some that appear to be classical, some that are a little bit more mixed. Now, you might say, well, why is this important? The answer is that it turns out to be prognostically valuable. Patients that have the more basal-like tumor have more aggressive disease and, succumb a little bit more quickly. What's interesting is that even though this has prognostic value, we don't very often think about whether or not it has any therapeutic relevance. And so we began to wonder, could we use RNA state to select drugs? Well, the problem, as you all know, is that RNA is actually this really squishy feature. It's integrative. It measures a lot of things that are going on. So it couples together DNA mutations and epigenetics and tissue architecture and interactions with the microenvironment. And even more problematically, we don't know what the reference is. We don't know what we should be trying to model. And even if we did model it, we don't know if it matters at all. So you might say, well, is this something that we don't have to worry about? Do the models already get the RNA states right? Well, we can go and we can look at models that we have locally, comparing what we'd see in primary tumors or metastatic tumors to what we have in cell lines and organoids. And what we see is at a genetic level, we do a great job. They're basically the same. We have the right number of KRAS mutants, P53, and so on and so forth. But when we go look at the RNA level, what we find is that actually we do a very bad job. We're very significantly different. We see that there's some classical-like tumors using a variety of different signatures, some basal-like tumors, some that have some other signatures that are probably, due to the fact that these are actually, very paucicellular with respect to the tumor burden. So there's not a lot of neoplastic cells, and so sometimes you get some confounding immune cells. But when we go look at the cell lines and the organoids, the cell lines are all basal, and the organoids, are basically classical with some other weird signatures going on here. So what this tells us is that we're not, representing these clinically relevant phenotypes that well, but also there's something interesting, which is the cell lines go one way and the organoids go the other way. So you might say to yourself, well, why is there this divergence that comes out of the models? Is it maybe that we're selecting for, a particular state when we grow out? Is it that they're plastic and they evolve? So if you're in 3 dimensions, it's different than if you're in 2 dimensions? Or is there something that's important about the microenvironment that we lose when we put these cells into a dish? Well, it may sound really simple, but we just decided we would do a very simple benchmarking experiment where we would take a small metastatic biopsy, isolate out some cells and make a matched model, and then try and compare them directly. So it might not seem like it, but it's a very simple experiment. Now, you're probably asking yourself, well, that's great, but how do you do all of this with an incredibly small biopsy? Well, as you all know, over the past several years, there have been these transformative advances that I mentioned that where we can now take these complex mixtures of cells, isolate individual cells in increasingly small devices, whether they're microfluidic chips or reverse emulsion droplets or microwells. Within those little chambers, perform molecular biology, for example, reverse transcription to capture all of the RNA, and then look at our cells as these points in this high dimensional space. So here I'm showing you cells in 3 dimensions, where each dimension is a gene. We're measuring 20,000 plus, and that's really hard to visualize. So like we reduce this down using these simple 2-dimensional representations, where each point is now a cell, and cells are grouped based upon similarity in high dimensional gene expression space. And then, we figure out what the cells are based upon unique expression. So we might say, these green cells up here, we think are T cells because they uniquely express TCR relative to others. The technology that we've really tried to push in this space, while using many of the others, are picowells. And that's because they enable us to work with incredibly small inputs. So with our partner in crime over at the Coke, Chris Love, Todd, Mark, and Travis have really pushed a way of taking some of the approaches that you probably are familiar with that rely upon reverse emulsion droplets and simplifying them using microwells. And so Chris's lab had sort of pioneered assays on these 1x3 cover slips that contain basically 100,000 50 micron by 50 micron by 50 micron boxes. And so you can basically think of this as a big ice cube tray. And what we do is we use this as a way of co-confining the same capture beads that you would see in DropSeq or in 10x, and then capturing cells. But the difference is we just deposit cells on the surface and let them settle using gravity. And I'll say that we're not the only people to use microwells. There's beautiful work done by Peter Sims and Guoji Guo, and all the techniques have slight differences in how they're approached, and so I'm happy to talk about them. But what's nice about the way we do it is that literally you just put the suspension on top, so you can work with just about anything. You can work with peripheral blood to identify, what's happening in hyperacute HIV infection, like Sam did. You can take pinch biopsies that are half the size of a poppy seed and compare regions that are inflamed and uninflamed in ulcerative colitis, like Jose did. Or you could even take fluids where you don't know that there are cells, like CSF, which Jay did, and compare pre and post immunotherapy to see what happens in people that respond. And just to show you how low we actually can go, the same nasopharyngeal swabs that you probably all have been using to test for COVID, rather than just using it to test for viral material, we can actually get cells off of that and collect them on the arrays, sequence them, and then figure out what's different between individuals that are COVID positive versus negative, and what's happening in the cells that are actually harboring virus that might be actionable. So what's nice is that because this is such a simple platform, this isn't something that only can be done in our lab. Actually, we've spent a lot of our time going out around the world, helping to do partnerships, applying this to a number of different devastating maladies, including, partnerships in Thailand, where you all are now, looking at malaria. And I'll just say, as an aside, that I helped to co-lead the equity working group of the Human Cell Atlas, and we deeply believe in enabling and empowering people to do these sorts of assays. And so, if you're interested in learning how to do this stuff and you're new to it, please reach out to me. I'd love to talk about how we can help get you set up and running. So let me move back to PDAC right now and just say, well, now we're in a place where you can imagine how we could take a very small biopsy, load some cells onto an array, and then take the remainder and seed our organoids and actually culture them over time and sequence them to see what's happening. And so what this does is it lets us actually create a reference in vivo map where we have both non-malignant cells and malignant cells, and get maps of our malignant cells from our organoids and compare them back and forth. So, the first thing we have to do is we have to figure out what the tumor cells look like. And it turns out that the tumors are a little bit more complicated than we would have expected in bulk. We see, much like people have seen in other cancers like Suva and Roche, that there's sort of this tripartite spectrum that emerges. So if we look at our spectrum as going from basal to classical, we find that there are cells that are classical and that are basal, but also some cells that co-express both programs and a couple of unique genes, which I'll call intermediate co-expressors. To make a long story short, I'll say that we can validate this using immunofluorescence, and that interestingly, when we look at tumors that are predominantly classical or predominantly basal, they have fewer of these cells than ones that are more equally mixed, suggesting that maybe these are a little plastic. Well, if we want to think about plasticity, we have to go to the models. And so what we can do now is we can go back and look at what's happening in our organoids over time. So we might first say, well, what do the organoids look like? Well, it turns out that they look pretty different than the biopsies. In our biopsies, we have cells that are basal, some that are co-expressor, some that are classical. In the organoids, we get some that look classical, but we also get all of this gene expression here that we don't see in the biopsy. And this really has to do with oxidative stress, amino acid metabolism, all the things that you might associate with the culture of cells. So imagine I now have 4 states: basal, co-expressor, classical, and organoid. How does this evolve? Well, what we can do is we can actually track these things over time and create these really complicated swimmer plots. And so what I'm doing here is I'm plotting pie charts that tell you about the fraction of cells that were basal, co-expressor, classical, or organoid specific. I'm telling you when we couldn't culture it out anymore. And this is really complicated, but I want to show you a couple things really quickly. I want you to see that, most of these things are evolving to being classical over time, but let me just give you a couple quick vignettes. So here in this intermediate co-expressor, what you can see is that over time, we go from being a co-expressor to being primarily classical. If we use copy number variation that we infer from the expression, we can tell that actually this is due to the rise of a new clone that seems to be classical in its phenotype. But what we see interestingly when we look at other examples is that even within the same copy number variant clone, that there is evidence of plasticity. So clones that are expressing basal genes at one passage can be expressing classical genes in the next passage, suggesting that actually the genetics do not lock you into a particular transcriptional state. But then again, there are places where the genetics are determinate. So here, when we gain a p63 amplification, we end up actually finding that it pushes our clone to be basal. And so we end up with a model that's basal overall. So in most places, things can drift, but sometimes the genetics locks it in. So you might say, well, what is it that's really maintaining basal phenotype? Well, to figure this out, we can go back and we can compare the organoids to the models, or the models to the biopsies one more time. And what we find is that, when we compare basal in vivo to what we would see in our model, that we're losing a lot of things that look like interferon signaling. And we can go through and do this for each of these. And what you discover is what's missing, what's changed is everything you'd expect. It's antigen presentation, it's extracellular matrix, it's focal adhesions, it's the things that you would normally see in vivo. And so you might say, well, is there something then specific about the microenvironment that associates with both of these, with all 3 of these programs? Well, we can go look at the non-malignant cells, and to make a long story short, it turns out there's a specific type of tumor-associated macrophage that's enriched in basal cells, or in basal tumors. It turns out that the basal tumors are less complex in terms of their diversity than the classical ones, that the intermediate co-expressors are strongly enriched for T cells, and that the classical ones that are diverse have a different type of TAM, these SVP1 high TAMs. So there is sort of an association between the microenvironment and our tumor state. So to summarize really quickly, what I've shown you so far is that the microenvironment can influence phenotype independent of genotype. That when we culture, we lose a lot of those interactions that are usually very important in vivo, and that there are links between the microenvironment and cancer cell state in vivo. So really, what we're saying is that we look at these, biopsies where normally we'd have autocrine and paracrine signaling, we strip them out, we add all these nice organoid factors to help things grow, and it leads to these strange drifts. So you might ask yourself, well, what happens if we just take out the organoid factors and we stop adding Wnt and R-spondins and TGF-beta inhibitors? Well, what you find is that actually the models go back to being basal and you start to lose the organoid state. It's still stable, it doesn't grow quite as fast. But what you find, just to really drive this home, is that if we have a model that's classical and we now go and remove the organoid factors, over time, the model will drift to recapitulating all of the states that we naturally saw in vivo. So we'll get basal, co-expressor, and classical. It turns out that cell lines that we've been growing for 30 years that are primarily basal, if we dump them into organoid media, we can make them classical. If we take organoids that are classical and put them in cell line media, we can make them basal. Now, this suggests that the microenvironment is important. What about all these autocrine and paracrine signals that would normally show up in the tumor? What happens if we start playing with them and adding them back? Well, this is a pretty complicated plot, but basically what I'm saying is these are factors that are higher in basal, higher in classical, higher in co-expressor. And so, for example, you see TGF-beta2 right here, suggesting that autocrine TGF-beta signaling from cancer cells is important for locking in sort of the co-expressor state and maybe a little bit more on the basal side. So the hypothesis is you dump in TGF-beta and you should be able to make this model, be a co-expressor. Turns out this works beautifully. You take your model, you add TGF-beta, and all of a sudden it goes exactly where you would have expected it. Now, you're probably saying to yourself, Alex, this is great that you figured out how to toggle back and forth this model. How do you know that it matters? Like, why are you telling me about this? And the answer is that it turns out to be incredibly significant. And so if we take the same isogenic organoid model and we put in the basal state or the classical state, and we add drugs that would be used normally in the clinic, what we find is that there are significant differences. So our classical models are much more sensitive to frontline chemotherapeutics like gemcitabine than our basal models. And you might say to yourself, well, isn't that just because the basal cells are more aggressive? And the answer is no. It turns out that the basal tumors are actually much more sensitive to other classes of drugs. And to really drive this home, what we can do is we can take one model, add TGF-beta, move it up to where I told you it was, keep it going, and it will get even tighter exactly where we would have expected. If we take a fraction of those cells and we remove TGF-beta, you see that it goes right back to where it was before. If we look at the drug responses, they're perfectly plastic. We go from being very sensitive to not sensitive with TGF-beta to sensitive once again, we strip it away. And what we can do is we can actually go look at a panel of drugs, and what we find is that when our models are in the classical state, they're much more sensitive to chemotherapeutics, whereas when in the basal state, they're much more sensitive to MAP kinase inhibitors, things targeting MEK and ERK. And so what's funny is that these are critical features, that we're missing when we're screening. We're not really thinking about state, but it turns out to be incredibly important. And, I think that this is a complicated way of saying something that is really simple, which is that mutations are important. But if we think about what a cell is, depending upon at a given moment, what it's trying to accomplish, what its state is, and we try and target that instead, we can sometimes have very effective outcomes. And then you might say, well, this isn't a dish. Does it turn out to be translationally relevant? Well, our data would predict that, taking chemotherapy would be much more effective for patients that have classical tumors than basal tumors. And that's exactly what you see when you go back and you analyze data, that was collected in previous clinical trials. So individuals with classical-like tumors have reductions in tumor volume, whereas those with basal-like tumors are the ones where you see growth. Now, this is all autocrine. What about paracrine? Well, remember, for example, that I told you that T cells were highest in co-expressors. You see gamma here. You know what I'm going to say. The hypothesis is I take gamma, I dump it on my model, it becomes a co-expressor. It works. So you generate this. You might say, well, does this happen in vivo? Is this something that actually is relevant? Well, it's hard, but we got lucky and we got one patient where we got a biopsy before immunotherapy and then after. And what you can see is that before treatment, the majority of the cells are classical. When we treat, we get co-expressor and basal expressing cells. It turns out that there's an increase in basal and interferon response genes, a reduction in classical, and that our T cells post-immunotherapy are really showing signatures of gamma activation. And so this is funny because actually what we've done by giving immunotherapy, this incredible treatment, is we force the tumor into a more aggressive state. And so, you wouldn't want to combine this with chemotherapy, but if you combined it with those MAP kinase inhibitors that I talked about previously, you could imagine this is a way of creating a more effective treatment, basically using immune pressure to control the state of the cell and then targeting it when it's vulnerable. So I hope what I showed you here is that, microenvironmental signals are critical regulators of transcriptional states and plasticity and PDAC, that we really need to think about the environment when we model to make sure that we create high fidelity systems for testing hypotheses. And that this, hopefully is a generally applicable framework. And so what I really just showed you is sort of a vertical stack in PDAC, where we tried to make in vivo reference maps, tried to create appropriate models, and then tried to identify state-specific therapies. Well, we tested a couple of intrinsic and extrinsic factors here. You might say to yourself, well, how do you scale this? How do you really figure out the full landscape? Well, Evelyn and Wall in the group, along with Julia Zhuang in Feng Zhang's group, have been trying to test the intrinsic side by adding in ORF libraries. And so here's an example where we add in, just a particular thing, that P63, that amplification that locked us in before. And you can see now when we add it that all of our models go up to being basal. And so what we're working on now is taking a large bar-coded human TF library, putting them into cell lines, and then trying to figure out which ones drive which state. And we're developing a couple clever ways of screening this at scale. The nice thing here, though, is that we have the barcode inside the TF. So we basically just need to figure out what's basal, what's classical, what's co-expressor, and then we can look for what's enriched in each state like you would with a normal screen. How can you do this for something that's cell extrinsic like interferon gamma? Well, this is where some work that Connor and Ben and Lab have been doing that's relatively crazy seems to be incredibly helpful. So they've been trying to develop this new method that we call compressed screening, where if you were to imagine a normal screening, say you had 8 compounds and you wanted 4 replicates, you'd need 32 samples. We began to wonder if we could pool together, drugs or ligands into random combinations, just making sure that we didn't get pairs of the same, or the same pairings that often, and do it this way in what we call a compressed screen. And then the idea is that we're just going to go do a simple, linear regression model to try and figure out what is the impact of each perturbation. And we're not the first to think of this. People in the pharmaceutical industry tried this a long time ago. It didn't work quite as well as they might have hoped. But we thought that maybe that was because they didn't have sort of the same ability to make a very high content measurement. So when you're just looking at something like cell growth, it might be harder than when you're looking at something like, in situ imaging of multiple transcripts or proteins, or doing single cell RNA sequencing. And just to show you that this isn't quite as crazy as it might seem, we can take those same PDAC organoids. We can take 68 of those ligands that we would have nominated from the environment and pool them together along with creating some reference compounds, do our sequencing, and then try and figure out what are the gene modules that are driven by each. And what we find is that 9 of them seem to drive specific programs. 3 drive an NF-κ for the activation pathway, IL-1 alpha, beta, TNF. We actually interestingly see a wound healing response that's distinct from what we would have seen in a canonical like IL-4 signature that comes in from IL-4 and 13. We get the TGF beta response plus another molecule in the TGF beta pathway and the gamma response. What we can do then is we can go do a subsequent screen to validate these in singlet. And what we find is that actually 8 out of the 9 turn out to validate exactly the way we would have expected. And we actually think that this last one right here that didn't work out is a pipetting error in the way in which we were setting up our reference plate originally. So, what's nice about this is you can now imagine how you would do a really complicated screen on a really complicated model, even in a small number of assays. And so, that's with biological ligands in a complicated model, but you can imagine how we could now start thinking about taking a lot of different drugs to see which ones might be the most effective in our models. And we've seen that even in highly biologically active libraries that we can get compression ratios of 40 or 80 or more at once. So that's going vertical. How do we go across? How do we think about other cancers? How do we really complete the square? Well, we've been trying to create some reference maps that would help us to think about other cancers, many of which have a basal-like state, which is usually more aggressive. And so Andrew and Mifra in the lab have been going through in combination with the NCI and creating single nucleus RNA sequencing data from a number of different TCGA samples. And so there's going to be a bunch of data that we'll be releasing soon that hopefully will be useful for you all. And I think you get the sense of where we're trying to go. We're trying to really move our way to completing the square, to understanding, sort of these pan-cancer ecosystems where we can think about, what's happening within them, what are the states, how do they depend upon the tumor microenvironment, to figuring out how to create the right states so that we can screen them for drugs. And then beyond that, not just thinking about, about models and their plasticity, but trying to think about how we could use surrounding cells within a tumor to control that plasticity to get more effective outcomes. Like I showed you with, the interferon gamma in PDAC and, the MAP kinase inhibitors. So within this is sort of the way in which our lab likes to do science. We like to tackle human biology, as messy as it is, of working with partners around the world. And this gives us good associations. But what we then need are appropriate models so that we can test those hypotheses so that we can accelerate interventions back into humans. And so we spend a lot of our time developing large libraries, of single-cell data, like many of you, along with Rosetta Stones that help us interpret, what we're seeing, those sort of perturbation data sets that help us go back and forth. And so, we've been standing up a number of different portals that bring together lots of our data across different diseases and models. We have a resource that we call Alexandria that James and Sarah have set up. It's called Alexandria for many bad reasons, primarily because my students wanted to give me a hard time. But what's nice about it is it gives us a way of generating hypotheses, that might be of relevance. So, for example, figuring out what would be likely targets of SARS-CoV-2 based upon ACE2 expression. But also it lets us figure out when we bring in a new data set, what might be similar to it so that we know where we need to look and, what would be a right place to test a hypothesis. And behind this, one of the biggest things that I think we need in the single cell field is we need information on multiple different systems. We need to understand what different compounds and cytokines and chemokines do, and not just at the RNA level, but at multiple different layers within the cell, because these are really the systems in which we test, and these are really the mechanisms by which we can push and pull. So with that, I want to thank my team. They're awesome, particularly all the people that were involved in the work that I showed today that are in hexagons here. But I want to thank everybody because it's really awesome to work with them. And I don't know why they work with me, but I appreciate it every day. And I want to thank, first and foremost, the patients and families. I don't think that we can ever thank the individuals that, work with us as collaborators in our studies by being donors. I think we owe them an incredible debt of gratitude, and that we, owe it to them to make sure that we do the best by them and by the data that they've enabled us to be part of collecting so that we can help others moving forward. I'll thank Bill, Andy, and Brian again, and all of our collaborators and our funders who've been very generous and hopefully will continue to be. And with that, I'll just say thank you and take any questions you might have. Thanks for having me.
Okay. Thank you very much, Alex. Thank you so much. It was a wonderful presentation and loved your work and your vision forward. I think we have some questions from the audience. So, we have time as well. So feel free to come up and just ask questions. Maybe Swain. Yep.
Okay. Hello. Hey. Alex, that was a beautiful and impressive talk. My name is Swain Chen. I'm at the Genome Institute of Singapore, as well as an Amazon scholar at Amazon Web Services. So I really enjoyed your talk. I think that example of being able to sort of modulate the sort of RNA phenotype that you were talking about with the PDACs was a great example to sort of illustrate the whole pipeline. But then, that had just ostensibly 2 states with an intermediate that then you guys were fortunately able to control, and obviously there's a lot of work that goes into that. So how generalizable is that? Right, so there's lots of other cancers that do have genetically driven subtypes that we know about, and I guess the question is there's lots of maybe other subtypes that we may not entirely know how that works, and so like how general is it that you could actually recapitulate that and control that in vitro, right, in a model, that then you could go and then do the whole testing of for looking for therapeutics?
So it's a fantastic question, and we don't know the answer, which is why we're trying to complete the square. I could tell you anecdotally that we've done a few other cancers and we've been able to see that we can do exactly the same thing. And the idea of controlling response in the way that I was telling you about by limiting plasticity, we've actually been able to do in leukemia, for example. So we can predict what are the pathways that we would try and target to make it so that the cancer be more sensitive to a targeted therapy like ponatinib, and we've been able to see that that's effective. I think that there are, some shared states. The basal state shows up in multiple different cancers, so I'm hopeful that that will work. but I don't know exactly how far we'll be able to take this. I'm 100% sure that there will be many places where the genetics are the most important thing. There will be places where the epigenetics are the most important, and places where RNA is useful. But we don't have a unified understanding of how it all links together, and I think that's why we have to sort of push forward. But a big part of this is making faithful models to states at the RNA level, at the DNA level, at the epigenetic level, because that will help us to actually address the question that you're asking right now. From the data that I've seen, I'm bullish that it may not be, 2 states, it might be 3 states, it might be 4 states, but in many of the places that we've looked, I think that actually it will be a more limited number than you might imagine, and that we'll be able to take advantage of this strategy, which is very akin to what people have done in the past in like a differentiation therapy as a way of creating better outcomes for patients. But hopefully, as more data comes online from us and from others, we'll get an idea of how broadly applicable these things are. And the last thing I'd say is just that we've seen similar concepts in other diseases outside of cancer. So I have reasons to believe that this may be a generally tractable policy. And just the back of the mind thing that comes to me in a lot of this is that if you think about it, our tissue ecosystems are only evolved to deal with so many threats. And so the idea that we would see recurrent themes that show up over and over again as a way of responding to them doesn't surprise me that much. Then the question becomes, how do the specific cancer states intersect with that? And that's what we really need to learn.
Okay, I had a quick follow-up, but I think you sort of answered it in as you were going along here in terms of like just this idea that, again, would it just be a linear thing? But you're sort of saying that some of them might actually have multiple states that might be on different axes that you would have to, in terms of the perturbations that you would do in vitro, right?
I think that there's that? So there's some examples of that in the context of like leukemia or also in glioblastoma. But I think that what you're really trying to do is you're trying to figure out how to control transition probabilities between states, and sometimes mapping out those hierarchies and trying to figure out what enables those transitions is really what we're after, because that's going to become critical to sort of controlling it. And I don't think there'll be one thing that's generalizable, but I think we'll be able to develop frameworks that will hopefully make inroads for a lot of people, if not for a large majority of them.
Great, thank you very much. Really enjoyed your talk.
Thank you.
Hello. Hi, Alex. My name is Sampath. I'm a former MIT graduate. It's really great work to see that the CRISPR chip, that I was actually there when that chip was designed, working with Joe Woman. Anyway, so my question regarding to your pancreatic cancer. It's very inspiring for me, but I'm trying to, when I see that work, what you just show there, I'm thinking about this palm tree where the different palm tree mutation of different cancer have different length and size and branching. So I'm curious on whether you have done any work trying to address that. But that is more from the, I guess, copy number gain loss and then the mutation pick up from the clone evolution. But I think you are approaching it from the more on the perturbation viewpoint and how it changed with the using organoid change with different drugs and growth factors, cytokines. So how so first question, how does this two, in your mind, kind of connect, and whether you have done any work that kind of may shed some light in that area? Thank you.
Sure. So it's a great question, and we're just starting to do some of it. So I showed you, for example, the one of the ORFs where we basically were trying to recreate the copy number variation that we had in P63, and that locked us into the basal state like we would have expected. The goal now is to look at a number of potential copy number variants by doing a larger ORF screen, across, over 1,000 transcription factors to figure out which ones are deterministic for cell state. In parallel, to think about losses, we can use some of the CRISPR, interference or perturbation strategies that, editing that show up. And so we're running those assays right now. We have done other things where we've tried to use different variants. And so there are some places where we have tried to toggle through, different mutated forms in the same way that we would do it with the ORFs and with the knockouts. But we don't have something generalizable there. And I think what we're still trying to understand is when do we need to mock the genetics perfectly? When do we need to mock the state perfectly? And we just don't know the answer yet. I think that this has really blown open a lot of questions for us. Like we would have expected some of these mutations, like I didn't talk about it because of time, but we would have expected certain KRAS mutants to actually force you into the basal-like state. It turns out that they don't. It turns out that, those are plastic. And it turns out that actually we find the highest activation of some of the RAS signaling in the intermediate co-expressors, the state that we didn't expect before. So it's early days, but hopefully we'll have more soon. But the way we'll do it is with knockdown and overexpression of molecules or their variants.
Thank you. Maybe I'll email you. Thank you.
Please do.
Maybe a quick question, quick answer. Yes.
Alex, this is Partha here. Hi.
Hey, Partha.
Was very impressed with this TGF-beta example that you gave. Reading between the lines, are you thinking that before initiating chemotherapy or some kind of drug therapy, you overexpress some genes to make those clones blow up and become responsive to drugs, and depending on the mutational profile, you then modulate which particular genes need to be overexpressed? Is that the whole idea?
You've got it perfectly, Partha. So in some places where the genetics are deterministic, it's probably easier because we know that the genetics lock into a state. So if we find the drug that the state is sensitive to, great. Otherwise, what we really want to do is we want to control the plasticity in the model by basically constraining the mechanisms of resistance or escape that can be used. We have a feeling that we can drive deeper remission. And so we've done this in a couple of cancers, but we're trying to think about it as a broader approach. And we think that not only can you do that by acting directly on the tumor cells, but that you can act on the tumor microenvironment as well by applying pressure from, cancer-associated fibroblasts or from, T cells or from myeloid cells, that we can think collectively about how we can constrain the way in which a cancer cell can respond to make it so that it isn't quite as fit in its environment. And that's what we're trying to work towards, a better understanding of how to do that holistically, if that makes sense.
Thank you.
Great. Alex, just quick question. How much does the sampling influence the profile? Because I could imagine biopsies are relatively small, but tumors can be much large. So
it's a great question, Jay. So we went back and we looked, I'll try and do this quickly, but we looked at some of the pancreatic biopsies and we did some spatial profiling, and it turns out that we didn't see things where it was confined to sort of islands of local microenvironment. Now, it could be that in the larger biopsies or, the larger resections that we had, that we didn't have sufficient tissue. But what we found was that, that we could predict what was going to happen in different compartments. So we found that actually in the lung metastasis, it looked different than in the pancreatic metastasis in exactly the way you would have expected based upon the surrounding cells. Mm-hmm. I think that what we don't understand that well quite yet is the length scale in our tissues, right? Because we don't understand the area over which, T cells survey, some of the cytokines and chemokines diffuse. And so there's a lot more for us to figure out there. I mean, and when I say us, I mean the field more broadly. Correct. Obviously, this is well beyond us, but we didn't see those little micro niches that we thought we might have based upon some of our things. It looked like it was more broadly distributed. And so it might be other things that are broader, like metabolism, et cetera, that play a role into this as well.
Great. Awesome. Thanks so much, Alex. You're quite late there, and we wish you were here in person, but hopefully next year. Well,
as you know, I have COVID right now, so
maybe not right now.
I think I'm not here in person.
Well, you're doing really well for someone who had COVID. But thanks so much for
sleep now.
Yeah, taking your time. Thanks so much. Bye-bye. Take care.
Bye.
Get well soon.
Thank you.